subdivided these neuronal populations. As I said, I want to figure out a way to interact selectively with the neuronal circuits that convey disease. For those of you who are more familiar with Parkinson's disease, you know that usually these interactions with neuronal circuits occur long after the cells have died. For example, if you're using deep brain electrodes, 80% of the dopaminergic cells have already been lost, and it's a less sort of a sub-effective band-aid to try and restore some normal healthy behaviors to a patient suffering from Parkinson's disease. And so myself and my collaborators at USC wanted to take a fundamentally different approach. That is, how can we promote cell health early on or interact with these cells before they are lost, before they have died in some degenerative process, so as to attenuate cell death and restore cell health. With this goal in mind, the tools that I mentioned are ideal for trying to stage disease progression, so you know at what point to interact with these neuronal circuits. To give you an idea of a degenerative process that has been notoriously hard to study. Not advancing now. And so let's say I was interested in studying something like bone degenerative disease. In this case, tissue clearing is actually quite effective in phenotyping the cells that you would like to interact with to restore, so as to prevent, for example, osteoporosis. In this case, the red cells are the osteoprogenitor cells, the hematopoietic stem cells. While many disease treatments have been developed to try and promote these cells within bone, it is very hard using traditional methods such as serial sectioning and random sampling of a bone to try and assess whether or not your therapy was effective. By clearing the entire bone, now you can very easily count these cell populations, especially at the interface between the bone marrow and compact bone, to see whether or not your therapy is effective at attenuating disease progression early on, as opposed to waiting until someone has very fragile bones. And so using this sort of teamed up some lovely investigators at USC to apply these methods to look at retinal degeneration. In this case, as I said, we would like to prevent the degeneration of cells in the retina, and in particular neurons within the retina, before the patient is blind. We want to promote their cell health rather than try and restore sight when there are no retinal ganglion cells left to convey a signal. And so in this case, we're using this simple ring electrode that's going to deliver an electric field to the eye, and we're going to look at, does that have an anti-apoptotic or sort of a pro-cell health to different cell populations of the retina across all 10 retinal layers, and then across the entire topography of the retina, not just looking at one little portion of the retina. This is obviously non-trivial to do. Traditional techniques, for example, thin sectioning, often are done in a small portion of the retina. You take a quick slice, you get very nice labeling, but again, you're looking at a small subset of the retina. And as I said, we want to look at the entire topography of the retina. You could do a flat mounting technique, which is quite common clinically, but again, the retina is actually pretty big. It's around 10 cell layers. So that means you are only looking at very superficial layers of the retina, even though the electric field is distributing across the retina. So we decided to apply a tissue clearing approach to try and be able to look at the entire topography of the retina and stage cell health versus cell death processes using both protein labeling and RNA labeling. Obviously, tissue clearing has been widely applied to the brain. The brain is fairly easy to work with. Mechanically, it's pretty solid. And several tissue clearing methods involving either dehydration with organic solvents, or, for example, hyperhydration have proven very effective with brain tissues and other more sort of solid tissues. The eye is a different can of worms, right? The eye is fairly fragile and has a 3D structure, and the other methods might not work so well. Instead, we wanted to use a clarity-based approach in which you would hydrogel embed the eye, so as to stabilize protein, RNA, as well as the 3D structure of the retinal. So to give you an example of this hydrogel embedding process within different organs, so this is some of my work from my postdoc, where I was using tissue clearing based on clarity, the return path, to essentially render all the different organs of a mouse transparent so that you could look at gene therapy approaches when you were using viral vector delivery. So now our job was to apply this to the eye. As I said, the eye is very different from other organs. And so we wanted to spend a lot of time in optimizing the hydrogel embedding process so as to stabilize the eye structure, as well as allow thorough antibody penetration and RNA probe penetration into the eye. As you can see, if you were to use no hydrogel to stabilize the eye, you get complete loss of the retinal structure. You can't decipher the different retinal layers. If you use a sort of minimal hydrogel, you can start to distinguish the retinal shape. You can start to observe the shape, but again, you don't get very good molecular phenotyping of the retina. And then we sort of landed upon a hydrogel formulation that we felt both preserved the 3D architecture, allowed probe, allowed visualization throughout the entire thickness of the rat retina. If you are familiar with the retina, you would say, well, several other tissue clearing protocols have actually been applied to eyes. Mostly they're applied to the mouse eye. Everyone loves their Thywan GFP mouse. It's very easy to validate almost any tissue clearing methodology with that mouse line. You also have other tissue clearing methods that applied to larger eyes. Typically, they're not doing RNA labeling or dual RNA and protein labeling, and they fall short. We actually did, though, try all these out, combining them with our hydrogel formulation. And in fact, trying out all the various tissue clearing protocols, we found that the cubic hyperhydration method, when combined with pack hydrogel embedding and a clarity-based mounting, they would give us a nice counterbalance between structural preservation, fluorescence intensity over time, and then the potential for RNA labeling. So I've been talking about the retina primarily because that's what we are concerned about. That has the 5 neuronal populations, all the other support cells, and in particular the retinal ganglion cells that project over to the brain. In degenerative models for eye disease, one of the most commonly used animal models, the RCS rat line. This is where a gene mutation causes degeneration and early loss of the RPE, the retinal pigment epithelium. And then it's this loss of support that leads to degeneration of the rods, cones, etc., throughout the retina. This means that we would love to keep the RPE intact along with the retina and potentially the rest of the eye. The RPE, everyone hates to image it because it is so autofluorescent. It is this big black smudge on the retina that you can see at the top. So while clearing and able to clear the retinal top, you are still left with the RPE layer that's going to block any laser trying to go through for imaging. So we inserted an additional bleaching step to essentially allow us to bleach the RPE layer, meaning that now we could perform this tissue clearing on the entire intact retinal cup with the RPE or on the entire intact eye and get very nice imaging. And I'll look at the entire time course of degeneration of when the RPE is still intact, but when it's quickly lost in rats, it's roughly 30 days, it starts to go pretty quickly, and then the retina starts to degenerate soon thereafter. So to give you just a quick idea, we are creating one image for the entire retinal cup using light sheet microscopy, and then we are diving in, taking out snapshots of various regions of the retina. And so this is simply one snapshot where we have labeled for GFAP and then a vasculature label, with these being two noted labels for degenerative processes that arise in the RCS model, where you see vasculature deform and increased gliosis across the retina. Okay. So as I said, so here's the digitally the final product is the retina right there, which hopefully you cannot see. And then we spent a lot of time ensuring that we had optimized the signal to noise ratio such that we could not only do protein labeling, as you saw on the previous slide, we could also extend this to direct RNA labeling using techniques such as single molecule FISH. But again, to return generation, here's an example of a wild type, and then the RCS and Ds model showing the generation. We are imaging the entire retinal cup of the eye, and then in the single image, we can dive in, start to quantify markers of degeneration or of the anatomy of the retina. And so using this, we sort of selected a couple markers we wanted to look at sort of off the bat to try and phenotype or stage degeneration so that we know the best time point at which to apply our therapy, in this case, electrical stimulation. So we'd be looking at things like remodeling of the retinal ganglion cells, gliosis, etc. So as a marker of gliosis, we can look at GFAP immunoreactivity, and we could sort of validate that this is functional. As I said, one of the generation, so you're only looking at very isolated portions of the retina. You might take a snap in the center of the retina, and when you do this, you get data. You see a change in, for example, an increase in gliosis, wild type, and RCS, as you would expect. And when you apply electrical stimulation, you see gliosis is attenuated ever so slightly, with electrical stimulation only applied to the right eye. So that difference between right and left being the effect of electrical stimulation. However, in performing behavioral tests of how well rats could see, essentially, we saw much greater protection than we expected, or than what this little chart would suggest, where there's not really a significance in the reduction in gliosis. And so we decided that we needed to take a more sensitive approach to phenotyping degeneration across the entire eye, since we had this nice, data set for the entire retina. So we automated the process of sampling across the retina at 4 points in the periphery and 4 points in the center. And then we also realized if we're going to start to do quantification, we need to normalize for the shape of the retina in the light sheet cuvette. It's going to be tilted in funny ways. Based on its orientation, we're going to see differences in the thickness of the layers of the retina, which is an important way to quantify disease progression. So we sort of did lots of normalization to correct for how the retina was oriented in our image. And now we can go in and pull out data in an automated fashion from the entire retina of each rat eye. The other markers of degeneration that we were interested in, obviously I don't have time to go over each of them, but to give you just a brief summary of some of the things we saw, that with stimulation would have attenuation of both rod and cone, and then you would have preservation of the thickness of each layer of the retina, of those 10 retinal layers, which is often used as sort of a marker of degeneration. And in particular, one of the things we were able to do now that we're able to get such massive amounts of data for both male and female rats, we saw that there was this very interesting selective enhanced protection conferred to female rats with electrical stimulation, showing that there is, in fact, not only sex differences in the generative process, but also sex differences in the response to therapeutic treatment, which is actually quite interesting. And so now some of the things that are currently underway are trying to look at the mechanism, how electrical stimulation might confer protection. To do this, as I said, I want some idea of cell health, metabolic processes, and activity, cell activity. And so we're doing things, for example, like connection labeling throughout the entire retina. So this is labeling for the gap junction. So we can now start to quantify gap junctions between photoreceptor cells and bipolar cells. And then again, the other way that we want to look at function is by doing RNA labeling. So we heard from the previous speaker of how she was using Visium spatial transcriptomics because she was essentially willing to sacrifice single cell resolution for doing whole genome discovery. In our case, we sort of want single cell resolution. We're going to quantify gap junctions on single cells, but we don't need whole genome resolution. We want to look at our 10 target genes or 100 target genes and get high quality information for that subset of RNA. And in this case, the tissue clearing methods that we've developed involving hydrogel embedding actually really help us perform these studies on large tissue samples such as the retina, where you're talking about several hundred microns for the thickness of the retina, but it has the 3D curvature. So now your sample size is millimeter scale. With the hydrogel embedding and tissue clearing, both lower the background, which is important for single molecule fish, and then you also separate the point labels so you can count them. In our first pass approach, we quickly found that the reagents and temperatures used for single molecule fish are compatible with the eye, even though they're compatible with the brain. However, we've since optimized the procedure to now being able to what we call our version of single molecule fish, so HCR, pioneered by Longpie. Labeling for various markers of interest within retinal neurons, and we're able to go in and count those. And again, this is in a very large image of the retina, and we can dive in and collect a ton of data. And so the elephant in the room, of course, as you might have guessed, is the size of these images. So for pop on a rat, it takes about 12 hours to acquire that image on a light sheet. The sort of post-processing takes about a week to 10 days, and you end up with obviously a very massive figure that you now have to work with. And so I'll leave it at that and say thank you for your time. I'm happy to take any questions.
Perfect timing. Maybe next in talk, does anyone have a question?
You mentioned 100 gene panel. I'm just wondering what is any ideas about how many genes you can track at the same time in 3D?
It'll be combinations of panels. So we're going to be looking at disease processes. So like, for example, the one I showed here, the PDE6A, that goes for the rod outer segment, present in the rod outer segment. Humans, as well as other animals that have mutation to this PDE6A gene, that is correlated with blindness.
It's not 100.
No, it'd be 10 genes at a time.
Oh.
So as I said, we are willing to sacrifice the whole genome scale or the 60,000 genes at a time for getting high quality single cell data that we have for the entire retina. Again, these are not 10 micron slices that we are reconstructing. This is RNA labeling across the entire retina, getting one image, and diving into count points.
I agree. It seems like you're working with quite a bit of data here. How are you exploring it, and what kind of structure challenges do you encounter?
Um, there are always some massive challenges. USC 2 years ago flooded their main server room, so there's a challenge. But we're storing it all on site. Luckily, USC has put a lot of money into computing recently. And the building where I am, the Michelson building in particular, is where the imaging center is. And it is wired for big data, but it's all storage on site. And it takes a long time for the data processing, right?
So I have a question. My lab is looking at Alzheimer's disease, and we look at brain and retina, and we know the challenges, at least by histology and biochemical analysis. Analyzing the retina is much more challenging than using the abundant of the brain. My question about the clearance is, when you talk about neurodegenerative diseases, obviously a key element is proteinopathies or protein aggregates. And so most of the clearing techniques would clear those protein aggregates, at least the extracellular ones. How do you see yourself overcoming this?
And so the cellular ones, you're banking on them being cross-linked into the hydrogel matrix. But the hydrogel embedding process, which is designed to cross-link with both protein, so the N-terminal protein, the side chains that have amine groups, we can cross-link those in to the extent that we can do labeling for connexin. So that gives us, some precedent. We are just starting to look at not just the RPE, but the debris zone that forms between the RPE and the first retinal layer, where you're going to have extracellular debris. And now we're trying to see how well can we label for that debris for random metabolic products within the debris zone. And we are getting some data. I don't know how much data we're losing because we can only see what forms in the degenerated rat.
Great.
Males don't know to be more colorblind. If I'm not mistaken, I think that's due to the fact that the colorblindness genes on the X chromosome. Does that have to relate with like males being more or less responsive to the treatment?
I think it's so we do the behavior testing to assay whether or not the rats start with reasonable vision, right? I think it's more related to hormonal effects. And so actually we paired with investigators. So I'm part of what's called the Center for Neuronal Longevity, which has some wonderful investigators, in particular, Dr. Louis, who does sort of mass spec proteomics work, and Baldur Sahia, who does single cell RNA-seq work. And so now we're starting to look at classic targets related to metabolism and the endocrine function to see if that could help explain some of the differences we see.
Our time is up, but if I may say, one thing that women has more micro, so they tend to have
they tend to
immune responses.
Yeah.
Than men. So that's one explanation of this neurodegeneration, because some of these activities or immunity contribute, exacerbate neurodegeneration.
Yeah, and so we see that in the female rat eye, gliosis much faster earlier on. Women get Alzheimer's disease, men get heart disease. That's sort of a fact of life. Thank you.